Hello everyone, today we are going to study about glycolysis. So let's start. Glycolysis is also called AMP pathway, Embden-Meyerhof and Parnas pathway. It takes place in cytoplasm. It is common in both aerobic and anaerobic respirations. In this process, in glycolysis, one molecule of glucose is converted into two molecules of pyruvic acid. It is a 10-step process in which all steps are reversible except 1/3 and 10th step, which is irreversible. There is no consumption of oxygen and no liberations of CO2 take place. So let's come to pathway. So in first step, glucose is converted into glucose 6-phosphate in the presence of hexokinase or glucokinase. Glucokinase in case of brain and muscles. In first step, 1 ATP is reduced to 1 ADP. And in second step, glucose 6-phosphate is converted into fructose 6-phosphate in the presence of phosphohexoisomerase. Since glucose is converted into fructose, they are isomer of each other. Now in third step, which is irreversible step, the first step is irreversible step and the third step is irreversible step, but and the 10th step is also irreversible, but all the other steps are reversible. So in third step, fructose 6-phosphate is converted into fructose 1,6-bisphosphate in the presence of phosphofructokinase. In third step, 1 ATP is reduced into 2 ADP. So basically 2 ATP are used here. 1 ATP are used in the first step and the second ATP are used in the third step. Now fourth step. In fourth step, fructose 1,6-bisphosphate is split into 3-phosphoglyceraldehyde and dihydroxyacetone phosphate in the presence of aldolase. So the first step, it's a split-in step, and in 5th step, the dihydroxyacetone phosphate is converted into 3-phosphoglyceraldehyde in the presence of isomerase. And in 6th step, 3-phosphoglyceraldehyde is converted into 1,3-bisphosphoglyceric acid in the presence of dehydrogenase. Here in the 6th step, 2 NAD plus is reduced into 2 NADH2. And in 7th step, 1,3-bisphosphoglyceraldehyde is converted into 3-phosphoglyceraldehyde in the presence of trans-phosphorylase. And here in 7th step, 2 ATP is used to form 2 ATP. So and in 8th step, 3-phosphoglyceraldehyde is converted into 2-phosphoglyceraldehyde in the presence of phosphoglyceromutase. And in 9th step, 2-phosphoglyceraldehyde, here the phosphorus is changing its positions from 3 to 2. 2-phosphoglyceraldehyde is in 9th step, 2-phosphoglyceraldehyde is converted into phosphoenolase pyruvate. 2-phosphoglyceraldehyde is converted into phosphoenol pyruvate in the presence of enolase. Here see, enolase is coming, enolate is coming, so the enzyme name is enolase. In 10th step, the phosphoenolase pyruvate is converted into pyruvic acid in the presence of pyruvic kinase. Here 2 ADP is converted into 2 ATP. So basically here 2 ATP and here 2 ATP. Total 4 ATP are forming, but here 2 ATP is used. So the net ATP will be 2 ATP. Now let's see the calculations of ATP. See, ATP directly produced is 4 ATP, but ATP used is 2 ATP. So the net is 2 ATP, but ATP produced via electron transport chain or electron transport system. Let's see. Here in the sixth step, 2 NADH2 is produced. So 2 NADH is produced. Since we know 1 NADH gives 3 ATP, so 2 NADH will give 6 ATP. So the total ATP produced in glycolysis is 2 ATP from here and the 6 ATP from here. So net total ATP produces 10 ATP. Thank you.